I don't think so, yeah.
Okay, thank you for coming. I wanted to introduce Phil Adamson. He's going to be giving his defense of his PhD work. Uh, Phil's worked with me for several years now. I forget when it started.
Something like that.
Has been really a fabulous member of the lab. Both you'll see some of the science. What you probably won't see is how active he's been in the Lucas community. So in addition to doing the research, he's very active in the educational programs we have. Particular one I particularly like is the one involving teaching science to in grade schools, and we're mostly in San Jose.
Yeah, second grade.
So it's been a real pleasure to work with Phil. You'll see we worked my part with some spectroscopy and metabolic imaging. During that time, he got interested in some of the newfangled stuff known as AI and machine learning, and Akshay was able to step in and fill the parts that I didn't know about. So he did the rest of his thesis both with me and with Akshay. And Akshay, you want to say something?
Yeah, so super excited to be here. It's been a huge pleasure working with Phil, like Dan described. You'll hear a lot about the research that Phil has done, but I think exactly like Dan mentioned, Phil has played such an incredible role in the broader RSL community. Phil started off his PhD in the throes of COVID, where I think really thinking about science and culture was really important. Phil has galvanized the community in the best way possible. He's been hosting so many events, bringing students and faculty together. So I think everyone in RSL really owes Phil a great deal of gratitude for all the time and effort that he's put in, not just to do fantastic science, but to build this amazing community that we're all a part of. So hopefully over the next 45 minutes or so, you'll get to see some of the science that he's been doing. And afterwards at 4 p.m. or so, hopefully we can celebrate the science as well as the impact that he's had on our community. So with that, I'll turn it over to Bill, please.
Well, thank you, Dan and Akshay, for the very kind introduction. So today I'm going to be presenting the work that I've done over the last 5 or so years during my PhD in electrical engineering on medical image acquisition and representation learning for precision oncology. So medical imaging plays a central role in the cancer care pipeline in that it gives doctors a window into the human body, allowing clinicians to probe underlying properties of cancer non-invasively and in a relatively safe way. This is done through various medical imaging machines and processes. For example, in MRI, there's an external magnetic field and radio frequency waves that probes underlying molecular spins that lets you see structure. This gives you, after the acquisitions, spatial frequencies, which you then reconstruct via a Fourier transform to get this MRI image. Understanding and making sense of these medical images relies on interpreting image features. For example, if you have a T2 FLAIR MRI scan, you see the bright spot in the bottom right of your picture. That may be some edema or swelling. In the lower image, there's a contrast-enhanced T1 scan with gadolinium, and some of the bright spots around the tumor represent places where there's blood-brain barrier breakdown in the tumor. Interpretation of these features is central, either by a radiologist or more recently by AI, and these features then inform clinical decisions. For example, has the tumor been growing or shrinking since the last exam? Thank you. Indicating disease progression, or is the patient stable, or does the patient need to alter their treatment plan if there is indeed disease progression? So today I'll be discussing 3 projects that I've worked on over the course of my PhD that span this medical imaging and precision oncology pipeline from image acquisition all the way to analysis and clinical decisions. The first project I'll discuss are deep feature distances, which are automated image quality metrics that give us confidence that the images that we acquire and reconstruct are of sufficient quality for clinical use. Next, I will discuss Merlin-Onc, a vision language foundation model for thoracic oncology, taking image features and translating them into improving clinical decision-making. Finally, I will discuss deuterium metabolic imaging, a novel acquisition technique that allows us to probe new features of cancer biology, for example, in this case, cancer metabolism, that other imaging modalities don't currently allow us to explore. But first, I'd like to do a brief intro into something that you may have seen in the title of the talk, deep feature representation learning. So you all may be familiar that if you go into the Photos app nowadays and you search for dog, it'll come up with every photo you've ever taken of a dog. And, this may be over 2,000 photos for some of us, or maybe fewer for others. But this obviously uses some form of AI. And how does this work? Well, it uses something called the deep learning classifier, where basically you have images of dogs, cats, people. You encode that image, and then there's a classifier that tells you if it's a dog, etc. These images or these models rely on layers, or essentially different mathematical features that take the image and convert it into something the model can understand. Early on in the network, these filters or mathematical operations take out things such as edges and sort of coarse shapes and colors, whereas later on in the network, you get sort of more abstract concepts as it combines these lower level features that it's learned. Let's take a look at the end of this network, where we have our model weights, the mathematical operations, then we have a classifier head to predict a dog. Right before that classifier head, there's sort of an important concept, which is known as deep features. This is what we're interested in here. And essentially, the model needs to use these deep features alone in order to predict whether or not the image is a dog. At this point, the image has been passed and converted into a lower dimensional representation, a lower dimensional vector, and all of the information that the model needs to use to determine if it's a dog is in this lower dimensional vector. That means that in a sense, this feature vector is a compressed representation of the image because it contains all the salient features that are needed to determine if it's a dog or a cat. So a deep feature representation is simply a learned via machine learning compressed representation of an image. So we have this feature vector, it's n dimensional, maybe 2,000, and we can plot this in a feature space. If this were in 2D, for example, we can just plot two of these. One of these features we call feature one, another one feature two, and we can just plot this like it's on an X and a Y axis. We then take a new image, and this lives somewhere else in feature space. In this case, because it's the same dog, it should live relatively close to the other image of the same dog in feature space. If we get an image of a new dog, it will be a little bit further away because the features are close but not quite the same. Whereas if we get an image of a cat, it should live somewhat far away. This allows us to draw a line in our feature space, separating dogs from cats. That's how classification works in this case. And it's not a coincidence that these features are linearly separable in deep feature space. In fact, that's exactly what we were training the deep features to do when we trained it in such a way that the deep features go through a classifier to linearly separate these. All right, with that background, I'll now jump into deep feature distances, where we again have our MRI acquisition. This is done through spatial frequencies, but all you need to know is eventually you take a Fourier transform and you get the image that will be interpreted by a radiologist. The quality of this image directly impacts analysis and interpretation of these image features, which inform clinical care. As anyone who's been in an MRI scan knows that it can be quite long and burdensome. And so there's been a lot of research in recent years to speed up MRI scans, make them go faster. This is known as accelerated MRI. And in a sense, you acquire less data, giving you a faster MRI scan. There is a trade-off, however, to acquiring less data. The image quality will go down. There's a trade-off between how much you acquire and the ultimate image quality. And what we need in order to quantify this trade-off are image quality metrics. What is the cost in terms of downstream clinical utility for making the scan go faster? Now, conventionally, image quality metrics operate in pixel space, or they operate on pixels. For example, the simplest one is the mean square error. You take an image, you just subtract another images. Every pixel is a pixel-wise subtraction. You then square it and sum them all up. That's the mean square error. There's also normalized mean square error, which sort of normalizes to the intensities in the image. Another common one is PSNR, which relates to the maximum value in the image, but ultimately they're all sort of a function of the mean square error. Now, we don't actually really care about the image quality metric. What we care about is how the image quality metric relates to downstream clinical utility, which means that we need another measure to see how well it correlates to that. And commonly what you do is called a radiologist reader study, where you have a radiologist look at a bunch of MRI scans and you ask them, what's the diagnostic quality? If you were to make a diagnosis on this image, is this of sufficient quality over a number of characteristics? And what you want is for this image quality metric to correlate. You just basically plot a line to the radiologist diagnostic image quality scores. If that's the case, you can use your image quality metric as a substitute, if you will, for diagnostic quality when you're developing algorithms. Unfortunately, most common image quality metrics don't actually correlate well with clinical utility. Here we have one of our metrics that's commonly used, peak signal to noise ratio, plotted against the radiologist diagnostic reader score on a scale of 1 to 9. And in short, there's a weak correlation, but it's not as strong as we might like. If you read papers that promote or rather demonstrate new techniques for how we should reconstruct MRI images, many of them will report improvements in these metrics, but we're really optimizing for the wrong thing. We're optimizing for a metric that is only weakly correlated to what we actually care about downstream. This motivates the need for improved image quality metrics for evaluating these accelerated or faster MRI image reconstructions. Okay, so back to deep feature spaces. Just like we have with dogs and cats, we can put medical images through these models and also plot them equivalently in a deep feature space. But in this case, instead of different pictures of dogs, we actually have different reconstructions of the same image. So in this case, this reference image in red is kind of the ground truth, or it's what the image would look like if you acquired all the data and you get a high quality image. This 4x acceleration basically means you undersample by a factor of 4, you go 4 times faster, and this should live somewhere else in feature space. And what you want for your feature space is, so a deep feature distance is simply the subtraction of these two vectors. It's basically the vector or the distance between your reference image and your accelerated image reconstruction. And what you want is that your higher quality reconstructions are closer to your reference image than a lower quality reconstruction. So if you have an image that's undersampled by a factor of 8, you'd expect this to be farther away from your reference image because it's less similar. A high quality reconstruction is highly similar to the reference. Okay, so that begs the question, we want this deep feature space. What's the best way to learn these image weights? Ultimately, the image weights are going to dictate what our feature space looks like, what features the model learns to be important for assessing this image quality. We can train a model on a classification task, even with dogs and cats if we wanted. This is actually a metric that was proposed in the natural image domain. It's called LPIPS. It basically computes a deep feature distance between two images using a model trained on ImageNet, basically dogs, cats, people, etc. So our question was, do deep feature distances trained on cats and dogs work well for medical images? And our intuition or hypothesis was no, probably not. So we should actually train models on medical domain tasks in order to compute medical domain specific deep feature distances. So one model we explored was a model trained on RAD ImageNet, at the time, an essentially radiology equivalent to ImageNet, where there are about 1.5 million medical images from MRIs to X-rays to CT scans across a wide variety of anatomies. We also wanted to see if we could train a model on MRI specifically. In this case, our reader study was actually done on knee MRIs. And so we wanted to know, what if we made the metric really curated to the features that we care about for this medical image acquisition, for this anatomy? And for that, we wanted to train on knee MRIs. Now, at the time, we didn't actually have good labels for these knee MRIs to train a classification model on. And so we used something called self-supervised learning. In this case, this is known as zero-field inpainting. You basically put these white squares all over the image. You cover up different parts of the image. And then you have a UNet model, or basically a deep learning model, predict what the actual knee should look like. And through this process, it sort of learns what knees tend to look like. It learns important features given the context of the surrounding knee to predict what's inside of the knee. Now, ultimately, we don't actually use these predicted images, but it's shown in the sort of the AI community that this is an effective way to learn good feature representations. This is also sort of how some of the early language models worked, where you would mask different words in a sentence and the model would predict what fills in those words. That's kind of a BERT style model for those who are familiar. All right, so we look at LPIPs, essentially our natural image domain features. We look at rad ImageNet feature distance trained on medical images, and we look at our knee MRI specific model. And we then conduct a reader study. This is sort of our ground truth. We want our metrics to correlate to the scores in this reader study. To do this, we take 50 knee MRI scans from the FAST MRI dataset. We have a variety of image reconstructions. So basically, we have scans that are accelerated by a factor of 2, a factor of 4, and a factor of 6. And then to reconstruct them, we use 2 different AI models. And the motivation for this is that the field of accelerated MRI reconstruction, even in industry these days, are sort of AI driven. And so we wanted the types of errors, the types of mistakes that these models make to be embedded in the correlations that we're looking for. We essentially wanted the errors to be realistic to what's going on today. A UNet is a bit simpler. An unrolled network is more similar to what's being done today and includes a data consistency term. But really, the goal of these reconstructions was to just get a range of image qualities over to which evaluate our metrics. We then asked 5 radiologists to evaluate the diagnostic quality of these knee MRI scans, specifically the diagnostic quality of the cartilage and meniscus, sort of an important piece of tissue in the knees that's commonly injured. We wanted to know, given these images, how well would you be able to diagnose a patient disease on these images? From a scale of 1 to 9, where 1 is completely non-diagnostic, 5 is okay, and 9 is perfect quality. So these are what some of those images look like. On the top are the 2x accelerations, so we use half the data. On the bottom, we use 1/6 of the data, and you'll generally notice, at least if you've looked at enough knee MRI scans, that the ones at top are a lot crisper, they're of higher quality, and the ones at the bottom have a bit more artifacts and weird lines that aren't actually corresponding to anatomy. If we then plot this on the bottom, on the x-axis, our radiologist diagnostic reader score versus one metric, say that normalized mean square error, we get a correlation coefficient. And again, we see that the correlation is a bit weak. And you'll also notice here, so the orange points tend to be to the left. Those are lower radiologist reader scores. Those were the scans which used less data, the 6x acceleration, and the blue used half the data, so more of the data, higher quality image, the reader scores are more to the right. We then look at one of our deep feature distances. So this was our MRI-based metric, which we call SSFD, and we see that the correlation coefficient of 0.85 is much greater than the normalized root square error coefficient of 0.64. The Spearman rank order correlation coefficient, think of it like R squared for people who are used to lines of best fit using regression in Excel, but this looks for just monotonic relationships. It doesn't necessarily have to be linear, but otherwise it's the same thing. So if we now plot all the metrics we looked at, peak signal to noise ratio, normalized root mean square error on the left, as well as SSIM, which is structural similarity index metric. It's one of the other most common metrics reported in literature. We compare those to our deep feature distances, LPIPs, SSFD, and rad ImageNet feature distance. And clearly the deep feature distances correlate more strongly with radiologists' perceived diagnostic quality than the conventional metrics. And they even approach inter-reader variability. So this means that if you have radiologist A, how well does that diagnostic score correlate to what radiologist B thinks? Essentially saying that these metrics approach how well a radiologist would do at assessing image quality. The surprising finding was that LPIPs, these ImageNet features, these pictures of dogs and cats, are actually just as good at predicting diagnostic quality as these medical domain specific features. This was certainly a surprising finding for us, given that medical imaging based pre-training shows advantages, as we'll see later in the talk. But But nonetheless, this is an important conclusion for the community because deep feature distances, even those trained on natural images, are improved metrics for evaluating image quality and help bridge the gap between algorithm development, which often doesn't have the resources to actually conduct a clinical study and instead just looks at these metrics, and brings the algorithms that perform best in a metric-based study closest to algorithms that would do best in a clinical study. Surprisingly, again, we find that these domain-specific or medical-specific deep feature distances don't actually outperform deep feature distances trained on natural images. I could speak at length about why that may be. I suppose, in short, these models are very good feature encoders and essentially feature extractors. And we actually find that you don't have to go all the way to the end of the network, the ones where the concepts or the features are most abstract, to get the best feature distances. They often use something in the middle. And in the middle, we more have features like edges and sort of coarse shapes. And often when evaluating MR image quality, we're looking for things like, is it blurry? Is it noisy? And these concepts are a bit more fundamental to even natural images than the more abstract shapes and medical images. But nonetheless, these are empirical findings after all. All right, so now I will move on to the second project, Merlin-Onc, a CT foundation model for thoracic oncology, which bridges the gap from image features to clinical decisions with AI. So just a bit of background on foundation models. So conventionally, deep learning models, at least 5 years ago, were designed for task-specific use cases. And these were trained via supervised learning, meaning that you had an image, you had a label, say you had a picture of a dog, you had a label as a dog, and you trained the model to learn that it's a dog. Or in the case of medical imaging, maybe you have a chest CT scan of a cancer patient, and you're trying to predict that, okay, this is a stage 2 non-small cell lung cancer. More recently, the field has been moving towards foundation models. These are designed for a wide range of use cases. They're trained on heterogeneous data via self-supervised learning. And we're all familiar with the general domain foundation models. There's ChatGPT, there's Gemini. These are sort of trained on a wide variety of tasks and aren't just meant for one task, but they sort of have broad knowledge. In the medical domain, there are some examples of this, MedGemma from Google, as well as Merlin from our lab, a CT foundation model. So again, we'll go back to our feature space. We have, in this case, a chest CT scan of a cancer patient. We embed it with some weights from a model that I'll touch on in a second. And we then get our feature representation, which we plot in feature space. In the case of Merlin-Onc, because this is a vision language model, which is trained via contrastive learning, text shares the same feature space. So we have CT reports. This sort of describes the findings in an image from a radiologist. Like this red circle here corresponds to a pulmonary nodule, basically a lung tumor. It passes through a text encoder. This is the equivalent to the image encoder, the image model that we saw before, but it's designed for text. We then get text embeddings. These are the same as our image features, but now we have text features. And the way this training works is that we put those text features in the same feature space as the image features, because we hope that there's a shared understanding between language and text, or language and images. So the way this works is we have CT reports that correspond to that image. So a patient took an MRI, a CT scan, the radiologist wrote a report for that image. That's called a positive pair. That image belongs to that report. During training, we also then have negative pairs. This is a radiology report from a different patient. This patient was completely healthy. Their radiology report says lungs normal. And we also pass that through a text encoder. We get deep features, and now we get the features in feature space in blue. Now, the way that contrastive learning works is it uses a clip loss. And the goal of the clip loss is to bring the positive pair, bring the image and text features that belong to the same patient closer together while pushing the negative pair away. Essentially learning that features of this image belong to the same features of this text, that this text is similar to this image. So we use this contrastive learning training technique. And we then used 15,000 chest CT studies from 4,500 thoracic oncology patients or lung cancer patients from Stanford. We trained with these radiology reports as well as ICD codes. These are like diagnosis billing codes that go to your insurance company of what disease can we charge for. We then train a model, the feature encoder for the CT images. It's a ResNet 152. It's basically a convolutional neural network. It's sort of similar to the encoders we were looking at before. And then the radiology reports uses clinical longformer. This is a BERT style model, unlike, say, ChatGPT. We then train with this clip loss as well as the ICD code loss and fine tune a prior Merlin abdominal CT model that was trained in our lab. This was a CT model trained on about 25,000 abdomen CT scans. So we now have our Merlin onc, and at this point, we're just looking at the image encoder. So we trained a text encoder too, but that was more to sort of shape the image features to have some correspondence to radiology disease concepts. But now we're sort of going to throw the text encoder away and just look at our image encoder. At this point, instead of looking at, say, different versions of the same image like we were doing in the reconstruction setting, we're looking at different patients. So patient 1 is passed through the Merlin onc image encoder. It lives over here. Patient 2 is over there, et cetera, patient 3. And what we're going to do is we want to now evaluate in some lightweight way, some easy way that we can do to assess, how good of a foundation is this of weights over features is this model learning? And we do that with essentially these linear classification tasks again. So for example, if we have a patient with different stages of cancer, say local disease, just like one tumor, regional metastases within the same organ, or distant metastases where it's like stage 4 cancer and it's spread throughout the body, what we want in this case is for these features to be linearly separable, just like we saw with dogs and cats. The distinction here is that in the case of dogs and cats, we were training the model specifically to linearly classify dogs and cats. What makes this a foundation model is that we're assessing these image features on tasks that are different from the tasks it was trained on. It was trained on sort of pulling and pushing different reports together. But what we hope is that the features that it learns about the image are sort of generally useful for oncology tasks. And so we assess in this talk 3 different oncology tasks. The first one is the cancer stage classification. So you take a diagnostic CT scan and you diagnose this stage 1, stage 2, et cetera. We also assess cancer progression assessment. So given a current and a prior CT scan, can we diagnose whether the cancer is worse than it was? So basically, a patient gets a scan at a month ago, and now they get a new scan after some treatment, and the doctor wants to know, is the cancer getting better? Is it getting worse? Do we need to change treatment? Are they doing well? Should we stay the course? This is known as cancer progression assessment. And finally, we predict 2-year survival. Will the patient be alive 2 years from now, given just this CT scan? And so here are some results for cancer staging. On the left, we have AUROC. This is essentially an accuracy metric. And on the right, we have AUPRC, another accuracy metric, specifically looking at precision versus recall. I guess all you need to know is it's 2 accuracy metrics, and higher is better. So in green, we have ImageNet. This is what we saw before. It's the same ResNet 152, but it's trained on ImageNet weights. Doesn't do very well at the task. We then have BioMedClip. This is a model from a couple years ago, trained on general biomedical tasks from Microsoft. We then have Merlin. So this was the original model trained on abdominal tasks in blue, or abdominal CT scans. And then we have our proposed model, Merlin onc in red, trained on thoracic CT scans. And generally what we see is that Merlin onc has about 10% improvement over Merlin at these tasks, meaning that it's learned better oncology-specific features. We then look at disease progression, and we see a similar trend. Merlin onc has AURC of about 0.66, 10% better than Merlin and better than the other 2. Same thing with the area under precision recall curve. And finally, we look at 2-year survival. So we have the same 4 as before, but now we've added a new one where the gray one on the right is a baseline prediction of survival that uses the disease stage, and demographics. So this is like tabular data. And what we find is that using a CT scan alone, not really trained for survival tasks, it never saw outcomes, but the imaging features are linearly separable in such a way that it can predict survival as well as knowing the stage and some demographics about the patient. So in short, we find that Merlin-Onc learns strong oncology-specific CT features and provides a foundation for improving clinical decisions in thoracic oncology. I'll say that the models would not be used in the clinic using those linear probes. But the idea of a foundation model is that it provides a base of understanding across a wide variety of oncology tasks to be used in the future. And we're continuing to improve this model as we go. And I think the exciting part about this is once you have these features, you can start combining it with features that are learned from other modalities. So say you have the patient's clinical history and text, that can get one feature representation. You have patient genetic data, you have patient pathology data. When the model understands images in feature space, it's a more tractable problem. It's a lot easier to start combining and having the model, having the AI learn sort of different correlations across imaging, across all these multimodal representations that would be very difficult for oncologists or sort of a multidisciplinary health team to do on their own. All right, finally, I will discuss deuterium metabolic imaging. So this is a new imaging acquisition technique to assess a new aspect of underlying cancer biology. So we've seen that medical imaging probes hidden cancer biology, and each of these medical imaging techniques sort of probes it in a different way based on the interaction of the medical imaging physics with the underlying biology. So a CT scan is basically an X-ray, but in 3 dimensions. And this probes different aspects of tissue density. MRI scans are magnetic fields interacting with water molecules using radio frequency waves, and it probes different aspects of tissue structure, sort of, how fluid is the structure? Is it really rigid in some dimension? Is it constrained? And MRI is very diverse, as we'll see, but at a high level, I'd say it probes tissue structure. Finally, another one that's very important in understanding cancer is called FDG PET. An FDG PET uses a radioactive isotope tied to a glucose molecule, a sugar molecule, and it is able to look at essentially how much sugar is this tumor consuming or how much energy is this tissue using. And it turns out that cancers, because they're growing very quickly, consume lots of energy. And so it's a convenient way to localize where tumors are and sort of which tumors are really active by looking at how much sugar they consume. All right. Now you can imagine that each of these different imaging modalities, because the physics is a bit different, learns or is able to probe different features. These features may be learned by AI, these features may be interpreted by a radiologist, but suffice it to say, we can only ever visualize features that the underlying physics was able to sort of pull out to begin with. These medical imaging features as they currently exist are only a subset of all the features we'd actually be interested in, of all the aspects of cancer biology that a clinician might want to use to inform clinical care. So if we can develop new acquisitions that learn new information about cancer, we can expand the arsenal of clinicians for learning more about the tumor to best inform treatments. One example of this is that cancers have altered glucose metabolism. They process sugar differently, and I'll step through this. So there's kind of 3 pathways I'll show here. The first one, healthy cells, normal cells, they go through, if anyone has taken some form of like biochemistry, they go through the TCA cycle. We can visualize this through spectroscopy on the right. This will be important in a minute. But when you go through the TCA cycle, you don't produce any lactate, but you do produce glutamate and glutamine. We'll just think of it as GLUX. Just think if GLUX is high, GLX, that means it's healthy tissue. We're going through the TCA cycle. We then have healthy cells, but without oxygen. Anyone who's gone for a run and gotten out of breath, and then you notice that your muscles are really sore, this has to do with lactate buildup. And that's because when there's no oxygen, we undergo anaerobic glycolysis, still producing energy, but it doesn't need oxygen, and we produce lactate. It turns out in cancer, they also produce lactate, even though there's oxygen present. This is known as the Warburg effect. It's called aerobic glycolysis as opposed to anaerobic glycolysis. And basically, in this setting, we're going to be looking at increases in lactate, but decreases in GLX, which is sort of indicative of this cancer metabolic phenotype of the Warburg effect. Why is this important? Well, the Warburg effect is sort of a biomarker for cancer, and there's actually drugs in development in active areas of research that are trying to sort of reverse this Warburg effect, whereby they hypothesize that the rate of growth of the tumor will slow down if we sort of are able to reverse this altered cancer metabolism, basically make the cancer metabolism more like healthy metabolism. Let's reverse the Warburg effect, right? So in order to understand if these drugs are working, we need to be able to measure the Warburg effect in vivo, which current medical imaging techniques can't do. FDG PET does something like this. It measures glucose uptake, how much sugar is the cancer consuming, but it doesn't tell us anything about the downstream metabolism. Is it producing lactate? Is it not producing lactate? There was a paper about 7 years ago from Yale that demonstrated deuterium metabolic imaging is directly able to measure the Warburg effect, basically producing a 3D map of lactate versus GLUX in the brain. All right, so how does deuterium metabolic imaging work? So conventional MRI detects spinning protons at some resonant frequency. Just picture this like a radio, you tune it to 127 megahertz, you pick up whatever signals there. That's how we're sort of probing, processing water molecules in a magnetic field. Deuterium is just a proton or a hydrogen with an extra neutron because it kind of has a different weight, essentially. It has a different frequency. It spins at a different rate. And so we have a different coil, a different radio antenna receiver that's going to be detecting signals at 19.6 megahertz, about almost an order of magnitude lower than what traditional MRI would use. And just like before, using these deuterium frequencies, we can do everything we can do with MRI, just at a different frequency. We acquire spatial frequencies, our K-space. We take a Fourier transform that gives us our brain MRI image. Now, in order to get chemical signals, well, if we acquire essentially each spatial frequency as a function of time and just let it decay, the Fourier transform of time is frequency. And it turns out if you design your pulse sequence correctly, this allows you to probe frequencies, giving you something like spectroscopy. We're able to look at the spectra for folks who took any chemistry. There's a variety of ways to probe these different chemicals, but essentially you can do this with MRI. And deuterium metabolic imaging. All right, so how do we conduct these studies? We try to track the metabolic products of orally ingested deuterated glucose with DMI. Patients drink about 60 grams of this deuterated glucose. It's completely safe. We usually add a bit of lemon. I think there's some people nodding their head who volunteered for my scans in the past. Thank you guys. The lemon has no medical implications. It just makes it a little more pleasant to drink. We then wait about 45 minutes. We wait for your body to metabolize the deuterated glucose because we want to look at sort of where did this sugar go? Did it produce healthy? Was it sort of healthy metabolism, healthy tissue that produced GLUX, or was it unhealthy and produced lactate? Our goal in this project was to implement deuterium metabolic imaging at 3 Tesla. 3 Tesla is one of the most common MRI scanners that's available in the US. The first paper was done at a different field strength, but we wanted to demonstrate that you could actually do this on MRI scanners that exist at hospitals across the country and around the world in order to hopefully eventually demonstrate that you can translate this technology and actually implement something like this in the clinic. So to do this, we had to implement a little bit of new hardware, thanks to the help of Ron. We had to filter the 19.6 megahertz gradient noise. Essentially, the MRI scanners weren't really designed to operate at this frequency, and so there's sort of different properties, and you need to filter out noise. We then built a coil. This is basically our radio antenna tuned to our new 19.6 megahertz frequency. And we acquire with that FID-CSI, where you sort of have K-space and then let it decay, and you're able to get the spectra. It's a 3D image, so each pixel in the image is a spectrum. So it's kind of a 4D image in that sense. This is 10 by 10 by 10 with about 2.5 centimeter spatial resolution. So it's more coarse than a normal MRI image. And we performed a number of pulse sequence experiments to sort of improve the SNR. In other words, improve the image quality given the time that we had in the scan. So now I'll share just a few results of sort of 2 exemplary patients of kind of the things that you can see at 3 Tesla with DMI. This is the patient with brain cancer referred to us by our oncologist collaborator. And this patient had brain cancer, but was relatively stable on treatment. There wasn't actually evidence of active disease clinically. So this is on the top left, kind of you get metabolic maps corresponding to each spectra. I'll actually just kind of show 2 spectra here quickly. The first one is the blue box here. So on the top left, we have an MRI scan. This is just like a slice through the brain. The blue box is like contralateral brain. It's the part of the brain that's healthy. And we see here, this is our flux, this peak corresponding to this frequency. This is where lactate would be. And the takeaway here is that relative to normal volunteers, this is a normal amount of glux to have, and there's basically no lactate. So this is indeed healthy tissue. Where the lesion is, in the red box, where the tumor was or is, we still don't see any lactate. The lactate peak is very small. It's kind of at the noise floor. We also see a decrease in glux. This essentially corresponds to just lower glucose uptake. So this corresponds to lower glucose uptake. I have no idea what that is. Okay, I'll just continue. Essentially, if you took an FDG PET scan, this would have reduced signal. There's just less sugar being metabolized at all in this tumor. All right, now let's take a look at a brain cancer patient. Oh, shit, that's my slide. All right, I just pocket dialed an alarm there. All right, I guess I'm coming up on time. All right. So this is a brain cancer patient with that disease that, according to our clinician, is behaving relatively aggressively. So they're not doing too well. The cancer is spreading. And again, this is our same sort of big picture view where on the top left, we have all our different metabolic maps. These are kind of images of each spectra. I'll step through again the different spectra. This is now, we see here a T2 image, sort of the tumors on the left-hand side. This blue box is on healthy part of the brain. This is kind of in the cerebellum, like down here. And again, we see our GLX peak at around 2.5 ppm and no lactate, indicating this is indeed healthy metabolism. Over here in the tumor, kind of the bright spot in the T2 flare image, we see decreased GLX and then increased lactate at about 1.3 ppm, just basically frequency. So this indicates that lactate is increased, perhaps the Warburg effect, abnormal metabolism. If we now look at the ratio of lactate to GLX plus lactate, this is basically the fraction of metabolism that's producing lactate, the fraction of metabolism that's abnormal. We can get a map. So this is like our 10 by 10 image, roughly. Each pixel corresponds to a point of the brain, but now we've sort of integrated these peaks, kind of gotten the amount of signal under each of those peaks, and then taken this ratio. And what we see is that there's increased lactate over GLX plus lactate right in the region where the tumor is, potentially indicative of the Warburg effect or abnormal cancer metabolism. So in this work, we demonstrated that deuterium metabolic imaging is indeed able to probe novel metabolic information in patients with brain cancer correlating to disease severity. And our implementation at 3 Tesla, which is widely available, provides a path to future clinical translation of this technique. All right, so in this work, I've presented 3 projects that span from image acquisition of DMI to unlock novel cancer biomarkers, to deep feature distances, to evaluate image quality, to Merlin-Onc, to take those image features and translate them into clinical decisions with AI. We can imagine here that, okay, how does deuterium metabolic imaging fit into this context. I presented it at the end, but it happens at the beginning. Well, you can imagine doing the same thing with DMI. Deuterium metabolic imaging will get you images. There's the need to evaluate the quality of these images. Radiologists would then evaluate image features. In this case, is there the Warburg effect? And you just can, integrate that with all the information we get from other medical imaging modalities to make a treatment decision. In this case, for this patient, the disease is aggressive, demonstrated the Warburg effect. We may need to think about new treatments. Looking towards the future, we have sort of our existing structural with MRI and CT, as well as metabolic with FDG PET and now DMI, ways to probe biological signals. But in the Spielman lab with Dan, we're actively exploring other ways to measure new metabolic information. We've been exploring an exciting drug in rat studies with a potential effective treatment for brain cancer. And the mechanism by which this drug works is sort of unknown to us, but we have some exciting signals looking at lipid metabolism. So lipid metabolism isn't something that is routinely measured, but we believe by the mechanism of the drug, if we can measure lipid metabolism, we may have a signal to tell us how well is this drug working. We're also looking at redox imaging. This is with essentially a novel T1 contrast agent that's able to look at the redox state. So there's sort of all these different ways that we can continue acquiring novel information about the underlying biology. Down at the other end, in analysis, Merlin is just the beginning of sort of our work in imaging models for thoracic oncology or oncology more broadly. In line with some of the novel biological signals, we actually have FDG PET data for these patients, which we plan to incorporate sort of towards the multimodal part of cancer care is really exciting, as well as adding temporal information. Right now, most of these foundation models that are state-of-the-art only operate on one image at a time, a CT scan taken yesterday. But really, cancer is a very dynamic process. It's important to understand the trajectory of these diseases. And so if we can train foundation models that understand these changes in time, they may be able to better predict the ways in which the patient is going. This also ties very closely into our work on deuterium metabolic imaging, where because deuterium sort of probes the rates of growth, we're looking to see, how does the deuterium signal as a function of time impact the patient trajectory. All right, so that's all for content. Here are some of my journal papers over the course of my PhD that I've been involved in, some of my conference abstracts. I'd like to thank my funding sources, the National Science Foundation Graduate Research Fellowship, the NIH and the ARPA-H, as well as Kate, my wife. We would also, like to thank all the patients and research participants whose data contributed to this work. I was just sort of casually, presenting someone else's brain cancer to you all as my work, but really this is a subject who took valuable time out of their life to no benefit of their own contribute to science, and I'm really grateful and humbled by that. So I'd like to thank them. I'd also like to thank all the fantastic mentors I've had at Stanford. Dan and Akshay, I was, I guess, reflecting on my father-in-law asked me what was the most surprising thing about my time at Stanford, and I think it was actually how much my advisors really cared about me and my career trajectory. And at times when I was struggling or had family needs, it was always, family comes first, make sure to take care of yourself. And at times when my interests in technology changed or research changed, always supportive of the next direction that I wanted to go. And so I'm really grateful for that. I'm also thankful for all the friends I've made here at RSL. On the top left, we have the SEED program, that outreach program, really fantastic group of people who were committed to outreach in a 6th grade classroom. All the folks in Akshay's lab, lots of fun times on Tahoe retreats and, bar trivia night. I'm just really thankful. I feel like we have such a good group of people, and it's RSL and the Amy Center are just a really nice group of folks who want to do good science together. And, yeah, go to a bar and on a Tuesday for trivia. I'm really thankful for the folks I've met here. I'd also like to thank all my friends from around the world. I'm lucky to have such good friends from high school to college, to today in the Bay Area, that I'm still in touch with so many of them, and really grateful for all the support over the years, and the Zoom calls, and the summer trips, and hope some of you guys are online, and I love you guys. I'd also like to thank my extended family. I was able to grow up with a lot of cousins and grandparents, and family vacations, and such a loving and warm, close-knit family that it's easy to take for granted when that's all you know, but I'm really fortunate for that, that I was able to grow up with that, and really shaped who I am today. I'd also like to thank the Ackley family, my California family, the in-laws, just my family, Granny Sue and Grandpa Bob. Love you guys. Steve, Marianne, Lucy, Charlie, Jordan. It was a big decision for me to move to California from the East Coast, moving away from that family that I love so much. And I couldn't have gotten luckier. I guess there's slide transitions. I couldn't have gotten luckier in finding another family so close to home. Just being able to do a Sunday dinner every week means the world to me. And you guys have been so supportive. Me entering a PhD program, and always my biggest fans. And I'm so lucky to have you guys. So thank you for all the support. I love you. I'd also like to thank my siblings, Jacob and Julia, and Katie, Jacob's girlfriend, who flew out here for this. It means the world to me that you guys would, come all the way here to watch a 45-minute presentation. And visit like a nephew or something. But yeah, you guys are the best. I love spending every Thanksgiving together, even though we're kind of spread out around the world. I feel like we're all still so close, and always support each other, and call each other out when we need to be called out, and love each other. And yeah, I'm really lucky to have you guys. Growing up as one of four was a lot of fun. Always numbers for 2 versus 2. I'd also like to thank Mom and Dad. You guys are such unbelievable mentors, and I'm just so grateful for, with the careers that you guys had, still finding time to take work off on a Friday to take us to the zoo, or leave work early to come to my baseball game, or drive me across the country when I move out to California. I'm so thankful that you guys are here. And yeah, I don't know. I couldn't have done this without you. I think I call you each like 2 times a week, and the more I call you, the worse I'm probably doing. So I'm thankful that you always pick up the phone, and I love you. And finally, to my beautiful wife, Kate. I couldn't have, yeah. You're just so supportive, to the point that I'm not supportive of myself, and you're just always reminding me of why I'm doing this, why I'm in a PhD program. I'm here to help people. I'm here to learn. Always my biggest advocate when I'm not feeling grateful, or I need to pick me up. You always just know the right thing to say. No matter how long the PhD was taking, if it took 4 years or 6 years, even if maybe you were rooting for 4, you never said so, and you just wanted me to be happy. I love you so much. You, as I said in my wedding vows, you always remind me to have a good time. Life is short, and enjoy the moments like this. And thank you, and I love you. All right, that's all. Thank you. All right, I guess take questions from the audience.
If you ask questions, please raise your hand.
Anyone can ask any questions. There's no stupid questions, whatever you want to. Anusha, yeah. Oh, 2, okay. Buster. Yeah, I thought that Buster was featured prominently enough in slides 3 through 15 that he didn't need to be thanked. But yes, obviously Buster. Oh, it was this. Oh, I can send you the website. It's like some like AI figure generation thing that I found. Student free trial. Yeah, I mean, it was better than ChatGPT. You could like edit figures. I don't know. You had to be very specific, and it took a lot of iterating. But yeah, saved me. It's better than drawing circles in PowerPoint. So
Questions about science?
yeah.
I think it's amazing how you've done everything from the acquisition, like inventing new imaging techniques, to actually the analysis. Does it, now that you've created or at least been part of the creation of a new image acquisition technique that gives qualitatively different images that actually have different semantic meaning to them, how do you think the foundation models and adaptation of the foundation models should sort of be upgraded or adapted to these new innovations in imaging? Because in MRI, you can do so many different types of looking images, but the foundation models are supposed to be able to understand everything, right? So do you have any thoughts on this open-ended question?
Yeah, I mean, I think to start, it's really challenging when developing a new acquisition technique to sort of probe what a foundation model might learn, because you have maybe 3 data sets and a year from now you'll have 7. And so it's not really conducive to that. But I think we'll definitely start learning about this with FDG PET. Whether or not you should train a foundation model that sort of takes both as inputs, whether you should train them separately. For Merlin specifically, the radiology reports actually describe kind of what's in all the imaging that was done at that time. And in the context of a PET CT, the radiology report may sort of fluently describe features that are in both, probably motivating training them all at the same time. So,
That makes sense.
Katie, yes.
For this technology accelerate, especially clinical trials that we deliver patients with kind of tumors or metastasizing diseases, by being able to extrapolate what the disease state will be rather than waiting to extend the pressure.
Yeah, I think on like the AI imaging side, there's huge interest in selecting patients for trials, knowing when a drug is working before it actually works. And there are folks that use basically CT-based image features, kind of radiomics, to predict the efficacy of treatment. It can also be these deep features, sort of another covariate in selecting clinical trial cohorts. So instead of just age and sex and stage of tumor to balance your clinical trial populations, if we have these image features and we know that they're predictive, should we split or should we use it as a covariate and cohort selection, where we actually sort of balance the deep feature distributions that we know may be predictive of something, in order to sort of not have biases that we only find out retrospectively when, like, oh, if we had split this in advance, then maybe we would have gotten better results. So yeah, I definitely think so. Dan?
Yeah, Phil, thanks so much. Also want to express my gratitude for the contributions you made to Marcelo and it's really great to have student members that do so much. So thank you for that. The question is about Merlin Monk. So the AUROC was approaching sort of 0.7 versus this number exactly. How do you distinguish between like inadequacy of the model versus insufficiency of the data? Like maybe the answer isn't in the images. And so if you're thinking ahead, how do you decide between model improvement or data?
Yeah, that's a good question. I think something that you can do is kind of like, I don't know, scaling laws, for example. If you have n data sets, let's sort of see how performance is going to improve as a function of the amount of data that we have. Does it look like it's plateauing? That may indicate that the task is really challenging. I think the real answer is probably just talking to clinicians. Like, how hard is this task for you? Is this something that you intuitively think should be in the image? Is there a reason to suspect that the answer to this question is in the image, or do you rely on, other clinical features? Do patients just come to you and it's a coin flip, it's 50-50 and we never have an idea? So probably more that. Translating that to numbers is difficult, but I think intuitively, what should the accuracy be of detecting an X-ray fracture versus what is the accuracy that we'd expect of 2-year survival prediction is very different. We know there's a million factors that impact all-cause mortality. You're going to have lower accuracy than fracture detection. So that's where I'd start.
Maybe we have time for one more quick question, if there is any.
Hi, Jacob, Phil's brother. It sounds like in your image analysis, you were optimizing for improved images for radiologist detection. Would you ever consider optimizing for AI image detection? So just bypassing what the radiologist finds significant there, just gathering data from what the AI algorithm at that point.
Totally, yeah. People have explored this. If you have like a pre-trained AI algorithm for detecting lung cancer, not only are people optimizing image reconstruction, but people are actually looking at like what data should we acquire. So data is kind of acquired in this spatial frequency domain. Can we like optimize the acquisition for some downstream AI model? I also think that while we were looking at diagnostic or radiologist diagnostic or perceived diagnostic quality, what we really care about is diagnostic accuracy. And it's really hard to get enough sample size to see how predictive is this metric of diagnostic accuracy, because you need radiologists to actually diagnose images over a wide number of disease types, et cetera. And so there are actually automated sort of image quality techniques in the natural image domain. Is this a cat or a dog? You give it some reconstructed image or denoised image of a cat or a dog. Does it still know it's a cat or a dog? And you can do something similar. And I think that's the next steps for this is if you have a bunch of pre-trained models that are maybe even used clinically that predict lung cancer, survival, et cetera, how well do those do across a wide range of reconstruction techniques? So yeah, you're hired.
Well, let's give Phil a big round of applause.